focus on viruses and will specifically study those viruses which infect bacteria. And those viruses are called bacteriophages. So first of all, we'll start with the structure of viruses. You know very well that every virus consists of two major parts. One part is the capsid, which is usually made up of proteins, and inside this capsid there is a nucleic acid. That nucleic acid may be RNA, may be DNA, or in case of DNA, that may be single-stranded or double-stranded. So related to our course of recombinant DNA technology, we'll specifically focus two important bacteriophages. One of the bacteriophages is Lambda Macrophage and the other is M13 bacteriophage. Lambda Macrophage is a head and tail type of bacteria, and M13 is a filamentous type. Okay, so here you can see the structure. The Lambda Macrophage is icosahedral, have a head which is icosahedral and has a tail. All this is made up of protein, and inside this head there is a filamentous, or you can say linear DNA. Linear DNA is packaged inside this head. On the other side, in M13 is basically a filamentous type. It basically looks like a filament, but inside this filamentous virus there is a circular DNA. So you can note some differences, like in Lambda Macrophage the DNA is linear, and in M13 the DNA is circular and single-stranded. In M13 the DNA is single-stranded, and in Lambda Macrophage the DNA is double-stranded. Yes. So now we learn that how the viruses infect the bacteria and complete their life cycle. So the infection cycle can be divided mainly into two types. One type of infection cycle is lytic infection cycle and the other is lysogenic. So first of all, lytic infection cycle. In lytic infection cycle, virus first of all attach at the surface of bacteria, and after the attachment it injects its DNA into the bacterial cells. Once the DNA is injected into the bacteria, it replicates itself, like it creates so many copies, like many hundred of copies of viral genome. And after the creation of these so many genome copies, this genome synthesizes capsid proteins and other related protein. So these genomes, viral genomes are available and viral capsids are available. So ultimately, these viral genomes are packaged into the viral capsid. So in this way, thousands of different phage particles or viral particles are produced, which cause the bursting of bacterial cell. So it causes the lysis of bacterial cell. So that is why this type of cycle is called lytic cycle. Because the host cell is damaged, the host cell is broken down, the host cell is lysed. That's why this type of viral infection cycle is called lytic infection cycle. As you can see that the bacterial cell is finally bursted. So second type of infection cycle is lysogenic cycle. In lysogenic life cycle, these bacterial cells are initially not bursted. So what happened? What happened is that the virus create a peaceful relationship with a bacteria. Here you can see that virus injects its DNA, but till so many generations, the viral genome become quiescent. It become very silent. How it becomes silent? That the viral genome inserts itself into the bacterial genome and just replicates. It doesn't transcribe or translate its proteins. So that is why it creates a peaceful relationship with the with the bacterial cells and keep on dividing. So that is why the bacterial cell keep on dividing, but it carries, those bacterial cell carries the viral genome, but at some stages. So this part basically called lysogenic life cycle. So viral genome remain inside the bacterial cells for many thousands of generations, and bacteria keep on dividing. But bacteria carry viral genome. But after thousands or more than thousand generation, what happened? That the lysogenic phages again convert to the lytic phages, meaning that they cause the breakdown of cell. So how it happened? The viral genome excises itself from the bacterial genome, and it replicates itself, and after replication it synthesizes so many capsids. It carry out transcription and translation and synthesize so many capsids, and ultimately this viral genome is packaged into those capsids. So the bursting of cells take place, but it's a delayed bursting, delayed lysis of bacterial cells. So the first type, which was lytic cycle, the cell which is infected, that breakdowns or that become lysed at the end. But in lysogenic life cycle, the bacterial cell doesn't lysed or broken down till so many generations, but at certain stages it could break down. So this is actually the major difference. Or another major difference between lytic and lysogenic life cycle is that once the DNA is injected into the bacteria, that DNA replicates itself. That doesn't I can go back to the previous slide. In the previous slide, you can see in the lytic infection cycle, you can see that DNA replicates itself, and right after the replication, immediately after the replication, transcription and translation take place, which result into the synthesis of so many capsids. So this is actually the distinguishing feature of lytic cycle, which doesn't happen in the lysogenic life cycle. In lysogenic life cycle, once the DNA of virus inject, it doesn't replicate. It doesn't replicate, or you can say that if it replicate, but it doesn't synthesize proteins. It doesn't transcribe or translates. So that is one of the major feature which exists in the lytic infection cycle, not in the lysogenic infection cycle. So lysogenic infection cycle could also be divided into two major types. One type which is found in the Lambda Macrophage. So this type of infection is caused by the Lambda phage, where virus infect the bacteria, and they live together for so many generations, and by the end, the virus causes the bursting of bacterial cell. While in the other type of lysogenic infection cycle, which is found in M13 bacteriophage, that is a different type of lysogenic life cycle. It is also a kind of peaceful relation, initially peaceful relationship. So how it happened that bacteria attached to the surface of virus attached to the surface of bacteria, and it injects its DNA into the bacteria. Once the viral DNA is into the bacteria, it set a very specific pace with the bacteria. Bacteria also keep on dividing, while on the other side, virus also replicate, transcribe and translate. So viruses also keep on going, on the other side, bacteria also keep on dividing by binary fission. So this is a very peaceful relation, and it keep on going for so many generations. So generally it is said that every bacterial cell produce or secrete almost thousand of viral particles by every bacteria. and after secreting these thousands of particles, that bacteria divides. So in this type of lysogenic infection cycle, bacterial cell lysis doesn't take place. While in the previous type of lysogenic life cycle, bacterial lysis takes place, while there is no lysis of bacteria in this type of lysogenic life cycle. So these are the two major type of lysogenic life cycle. One type where bacterial cells break down, while the other lysogenic life cycle is where the bacterial cell doesn't lysed, and the viral particles are produced by every generation, by every bacteria, and these viruses are secreted from the bacterial body. Okay, so after that, now we'll briefly discuss about the genome of Lambda bacteriophage and M13 bacteriophage. As I have already told you that Lambda bacteriophage genome is almost 49 kb. So here you can see a map of Lambda bacteriophage genome. It's here you can see that it's 49 kb, and these are the different genes which scientists found that which scientists have reported that they exist on the DNA. So here you can see. So what we want to why the genome, why it is important to study the genome of these two bacteria. We want to know that what different pattern exists. So one important pattern is gene clustering in Lambda bacteriophage. Gene clustering means that some specific type of genes are together. Like here you can see the big concept of clustering. From 1 to 21 kb, you can see that all the genes are related to the capsid formation. So first almost 20 kb is related to the capsid formation, while here you can also see that there are some genes which are involved in the integration and excision, because you know that lambda DNA integrated itself into the bacterial genome. So these actually genes synthesize some proteins which help the viral genome to integrate into the bacterial genome, and there are some proteins are also coded by this region which are involved in the excision of viral genome from the bacterial genome. So in this portion there are the genes which are related to the integration and excision. Here you can see the early regulation related genes. So clustering of gene is very important idea which you should know, that in Lambda bacteriophage the genes are clustered, which means that same type or the genes which are related to each other, they are usually clustered. Like here with the capsid genes are in the first 20 kb, and some other genes of same activity are also together. Okay. Second important feature related to the Lambda bacteriophage genome is the cause site. Second important thing which you should know, that is all about the cause site. The Lambda genome have cause sites. What are cause sites? Cause sites are basically the single stretch of DNA of 12 nucleotides. So here you can see the cause site. This is basically cause site, and these cause sites exist at both ends of the linear DNA of Lambda genome. So they exist on both sides. This is the 49 kb bacterial genome or viral genome, and these are cause sites on both sides. So what is the function of these cause sites? The function of these cause sites is to make the DNA is to circularize the DNA and help the DNA to replicate. The second important function of cause site is that it is involved in the cleavage and packaging. Which means that if these cause sites are not available on the DNA of viruses, that DNA cannot be packaged into the capsids. So these are basically the signals. This is basically address to go into the capsid. So these cause sites are very important. If this cause site doesn't exist on any DNA fragment, that DNA fragment cannot packaged into the capsid. So they have very important role in packaging. So these two things are very important, which are related to the bacteriophage genome. The first part was gene clustering, and second part was importance of cause sites. Now a few words about M13 bacteriophage. You know, M13 bacteriophage basically is a 6407 nucleotide, and it is single stranded. So once it get into the bacterial cell, it circularizes itself. It circularizes or duplicates itself, and finally replicates. So this genome of M13 is basically a small genome, and it has large capacity to take a foreign gene of interest. So that is why this bacteriophage is very important in recombinant DNA technology, because it is very small size, it is easy to handle, and it has large capacity to take foreign DNA of interest. So this was all about the genome of different bacteriophage. This is just last slide, and we superficially want to go through this slide, which is about that what are the other viruses which are used for eukaryotic organism as a vector. So rhinoviruses are used as a vector in human. Baculoviruses are used as a vector in insect, and colimovirus and Gemini viruses are used as cloning vector in plants. So this was all about our lecture on viruses, its structure, its life, its infection cycle, its genome importance, and some other type of factors. So thank you very much. See you soon.